Pleasure to introduce Dr. Avi Mayan. He's a Mount Sinai endowed professor in bioinformatics, professor in the Department of Pharmacological Science, and director of the Mount Sinai Center for Bioinformatics. And you're gonna see that, I don't know if he's gonna show, but his lab developed several user-friendly tools that are very useful for many people around the world, including my lab. So we always receive it in the WhatsApp group, someone saying like, look what Mayan is the Mayan form of Brazilian saying, just publish. And then everyone tries to look at it. So that's why I'm so happy to be the chair of this talk. And Avi, thank you so much for accepting this seminar to give this talk.
Thank you so much for the kind presentation. I'm trying to share my screen.
Miguel, I think you have to authorize.
Yeah, if you can authorize me to share my screen. Okay. Yeah, looks like it's working. So you can all see my slides, the title slide.
It's just starting.
Yeah, not yet.
Oh, yeah.
Now it's on.
Okay. So I'm going to talk to you about machine learning and how we are starting to impute knowledge about drugs and genes in systems biology and systems pharmacology. And I'm going to start with this slide that showed that we are still in the dark in biology and biomedical research. There is still a lot of unknowns. We don't really understand how cells work, how the cells are structured, what proteins and genes are doing, how drugs affect cells and individual small molecules, and we have a lot of data that has been collected. So that is very rapidly increasing in size with all of those new biotechnologies. So there is a deluge of data, but only a part of that data is actually exploited for knowledge and full understanding. And there's still a lot of room for collecting additional data. So we still need probably a lot more data to really get to this point where we fully understand what happens inside cells and how their cells are affected by perturbations and how the whole system is working. So it's a very complex systems that we can't observe in a microscope. And that's why we really still are mostly in the dark in biology still today. So all of this data that is being collected is served on many of those databases that many of them are primary databases that just post the data, make the data available for the community. And this is just some logos about some of the widely used resources. And some of this data is organized in gene landing pages. So you have those resources that have gene cards, but not just gene cards, which is one of those databases. It's all of those databases that you see here, either host dedicated page for gene or protein or drug. So they're also drug landing page. For example, PubChem is probably the most used drug resource where you go there and you can find information about small molecules and their structure and other things that are known about either the drug or the gene.
Dr. Avi, I was informed that there is something with the link. Tiago, em português, você está aí. I think that the link changed a little bit for the meeting. So I'm trying to figure how to quickly fix that. Tiago, tá ouvindo, Tiago?
Oi, tô aqui, professor.
Parece que o link mudou, as pessoas não estão conseguindo entrar.
Eu estou recebendo aqui alguns e-mails, já estou olhando isso aqui, estou respondendo o pessoal por e-mail, tá? Já com o link certo aí para eles, tá bom?
Não, o link mudou mesmo? Me passa
não,
ele...
Na verdade, não teve alteração do link, assim, então...
Tá.
Alguém
Parece...
falou... Tudo certo.
Alguém disse que tem um limite de 50 participantes.
Ah, tá. Gente, eu vou dar uma olhada. De qualquer modo, quem está mandando e-mail, eu estou passando o link do YouTube, tá bom?
Tá, passa para mim, para nós aqui na audiência, no chat, que a gente ajuda também. Mas
Aí
o
o link do YouTube também.
não. Tá, mas você não tem o link à sala? Eu acho que quando corrigiu o acento, trocou alguma coisa.
Não, o link, o acento não chegou a ser corrigido lá não, professor, porque a sala já estava aberta, né? Aí ele não permite. Então,
não, tá bom. O limite é de 50, né? Tá bom, ok.
Isso.
Okay, Dr. Avi, we also have the option for people to reach by YouTube, so the audience is listening to you already. Thank you.
Oh, no problem. Yeah, I saw that there is a YouTube live broadcast. So this is listing of 43 different gene landing pages resources that we found. And if you know about additional ones that we missed, please let us know. But this is a pretty comprehensive list of those resources that have those landing pages. And we also, it's stuck for me. Collected information about drug landing pages. So these are 15 resources that have drug landing pages. And what we've done, we took the information from all of those resources and created this search engine that is not published yet. And it's an aggregator of gene and drug landing pages. So you come to this website and you can put in your gene of interest or your drug of interest, and then it presents you with cards that have information about each of those resources with an external link to view the information about the gene or the drug. So this is just a relatively simple application that potentially can consolidate a lot of the information about genes and drugs, and it's available on this URL. So this is a new resource that we developed. So all of this information that is coming from those databases can be organized. And this is a figure from a publication from 2014 that shows how you can start having those associations between genes, proteins, and also small molecules with other entities like side effects, diseases, tissues, mutations, and other type of like signatures and pathways. So all of this data, all this information can be represented as bipartite relationships. And we already thought about this in 2014. Now there is, in bioinformatics, there is a resurgence or realization that you can do this, and people are building knowledge graphs from this type of data to discover associations that are not obvious, but are still associations that are established based on experiments and the literature. Another figure from that review article from 2014 identified 7 different categories of data that can be abstracted into those networks or those bipartite associations, but also into Geneset libraries. And that's something that we are famous for, is converting many resources into Geneset libraries. You have a label and then a bunch of genes associated with a specific pathway or some type of phenotype. And here you have the categories, the 7 categories: the gene signatures, transcription factor targets based on ChIP-seq, effects of drugs on cells, cell viability type of data, knockout and mutations associated with phenotypes, gene expression, basal gene expression in patient cohorts in different tissues and organs, like the GTEx project, and then protein-protein interactions and metabolic pathways, cell signaling pathways, databases, and then the last one is the drugs and side effects and adverse events type of associations. So all of those types of data can be combined when you abstract it at the gene or drug level. So this is a high-level view of the type of entities that we deal with. So all of those objects like cells and tissues, genes and proteins, drugs, small molecules, gene sets and pathways, they all can be connected to each other based on those bipartite relationship. So one of the things that we've done was to create this database called Harmonizome, where we serve all this data, all this basically we went to all of those databases and we abstracted the data into associations between genes and their attributes. And this is covering 66 resources and generating 114 data sets that provide those associations, and some of those associations are weighted. And then once you come to this Harmonizome website, you can type in your gene of interest and you can get information about the gene or those other entities, but mostly the gene landing pages, like I mentioned before, that we have one of those resources that have those gene landing pages that aggregate information from so many resources. So this is the Harmonizome gene pages. So this is a gene page for the gene Klotho, which I'm interested in because it's an important gene that is downregulated in aging and is highly expressed in the kidney. And this is just the top of the page. If you scroll down the page, you get all of the associations for this gene in all of those resources. And I'll get back to this page later. So the Harmonizome in general, it has been widely used. It was a big success. You can see that it currently has over 900,000 unique users since it was published in 2015. And there is an increase in activity on the website, even though we have not significantly updated these resources since it was born. And there are also a lot of users from Brazil. So that's somehow because the workshops and the relationship that we had with the São Paulo University, we get a lot of users from Brazil, and we are very appreciated that relationship in general. We also have specific inquiries about how to use the tools, and we have been grateful for that. So one of those other very successful tools that does basically very similar type of analysis and approach to this is Enricher. And I'm not going to talk about Enricher in this presentation, assuming that most people are already familiar with Enricher. Enricher had over 40 million submissions of gene sets to the tool. And here you put in your gene set and then you find other gene sets that are matching your query of gene set. I'm going to focus today on machine learning and predicting knowledge, new knowledge based on existing knowledge. And this is this entry that we added to Harmonizome, which is 3 links to the Arches 4 resource. So if you click on this predicted functions for Arches 4, you get predicted functions for Klotho in the Arches 4 database. So a little bit about Arches 4. What is Arches 4? It is a project that started where we wanted to process data from GEO for generating signatures. And GEO, the Gene Expression Omnibus resource that have all of the microarray and RNA-seq data from the world, does not have these RNA-seq samples uniformly available. Basically, all the RNA-seq data is available through FASTQ files. So Alexander Lachman, assistant professor in my lab, he created this cloud-based alignment system where he's grabbing the files from GEO and uniformly align them with the Callisto aligner, and then make the tables of expression available through the Arches 4 website. and also through a Chrome extension. And one of the nice things that he did, he created this interactive 3D t-SNE visualization where all of the samples, either human or mouse, can be searched against and visualized. So you can put in, for example, a term like long, and then get all of the samples that mention the term long. And then you can download that subset of samples and do analysis on that. And this is a rotatable 3D visualization of all of the samples. Each point is a sample. So the Arches 4, in addition to this interface, has gene landing pages similar to Harmonizome. But here the difference is that those are predictions. Those are predictions for gene functions. For example, predicted human phenotypes for Clotho. So the way we are visualizing this data, for example, Clotho already has a bunch of known phenotypes in HPO. And then we see how well we rank those phenotypes. And this is just showing you that there is a ROC curve with a 79% accuracy of predicting terms that are associated with Clotho already. And this is all based on the Arches 4 RNA-seq co-expression data. So if you think about the Arches 4 resource, so this is a little bit about more details of how we make those predictions. You have the genes, all of the human genes as the rows, and then their expression across almost 1 million samples as the columns. And then you can take all this data and you can convert it into a gene-gene similarity matrix. So now we have the distance between the genes, and then we can use that distance to make predictions. So the way it works, for example, if you think about the first term, increase circulating renin level, which is the most predicted human phenotypes for Clotho. That means that the genes that are already known to be associated with phenotype are mostly correlated with Clotho. So on average, if you take all of those genes in that term and you compute their correlation with Clotho, you get the highest average correlation for those genes. So this is a ranked list of terms based on their correlation to Clotho, and those are predictions. Very simple way to do it. But you can do it in a more complicated way. You can start building machine learning pipelines that use multiple resources of data, and you can concatenate those data sets. So you can use expression, either basal expression in tissues or signatures, and then you can combine that with proteomics data, literature data, and then you can build different functions or prediction algorithms that you can put in to make predictions about whatever you want to predict. For example, here's want to predict a mouse phenotype. Let's say there's some genes that if you knock them in a mouse, the mice live longer, like by, let's say, 50%. And then you, those genes, those unique genes are observed for very few genes, but there's a lot of genes that phenotype is not observed. But there is also still about 15,000 genes that haven't been knocked out in a mouse yet. So you could make predictions about, from those 15,000 additional genes that haven't been knocked out, which one would give us the phenotype that we care about. And this can be done now with many libraries and tools that are available. For example, scikit-learn or TensorFlow, and you can test different machine learning methods. But the real important thing is the setup. How do you set up your problem? How do you pick your data sets? How do you filter and find the best features to make the best predictions? How you are not doing overfitting? And you can also implement things like deep learning now. And it depends on the type of problem that you have. You have to select the right algorithm to give you the best results. So the same thing that I showed you with genes, where you put the genes as the rows, and then you concatenate different data sets as the columns, can be done with drugs. So in this particular paper from 2016, we attempted to predict side effects using 3 types of data: morphological features that are captured with microscopy from over 20,000 drugs that are used to treat cells. So this is the data from the Broad Institute, as well as gene expression from the L1000 assay. And here you have 979 landmark genes that are measured, and then you have those as features. And also you can combine it with the molecular fragments of a drug. And then all of those features can be concatenated to build this classifier. In this case, it's a multi-label classifier that predicts adverse events. So there are over 1,000 adverse events that are listed either on the drug inserts or are reported after the drug is marketed. And you can use all of those adverse event tables as your class labels. And then you can, in this particular case, it was a random forest classifier. You can build a classifier to try to predict adverse events for small molecules that haven't been tested in humans yet. And this can give you a clue about what potential things a regulatory agency like the FDA should watch for when they receive the new drugs, as long as there is data about their expression and morphological changes. So this is done pretty successfully. And more recently, we published this database called Monizome, which is a Harmonizome version for drugs. And in this particular case, we did the same thing. We went to all of those resources that have information about drugs, and then we converted those to drug set libraries. And currently we have over 10 million associations between drugs and their attributes, and those are organized in 110,000 drug sets. So those are also served on the Monizome website, and you can do drug enrichment analysis instead of gene set enrichment analysis. So one of the recent papers basically going through the same exercise of making predictions based on combining some of those data sets. So this is an example of how you can do it. And this is a project that was led by 2 postdocs in the lab, Minjin Jian and Kathleen Jagodnik. And the first step was to actually find genes that are associated with pain. So we found that pain, is not just one, disease or condition. There are very different types of pain. And Kathleen identified 17 types of pain. And she used the pain-specific resources as well as general purpose gene phenotype or gene disease association databases listed here to establish gene sets that are associated with each of those pain types. So this is a table of the pain types. So you can also have a composite of all the pain types, which is over 5,000 genes. And then those other pain types, they have established genes that are known to have something to do with that pain type. And then once you have this table of positives, you can start building this model of machine learning model by combining different data sets. So the Minji took the positives of each of those pain types, and she took transcriptomic data, proteomics data so transcriptomics, she took the Arches 4 data, and she converted it to a co-expression matrix. And then she went to proteomics DB, and also took data from CCLE, which is over 1,000 cancer cell lines profile proteomics. And then she created a 7,780 by 7,700 by 80 matrix of protein-protein correlation. And then also she took the gene ontology tree and she made a Go similarity matrix based on how genes share Go terms. So now you have those three sources of data. You can combine them and build a model that uses each of those data sets similarly to what I showed before. It's an ensemble model, and she used the deep neural networks to use those features to predict the pain types based on the positives and the negatives that Kathleen generated. And the model is performing pretty well for some pain types, better than others. And the top one is arthritis. And what it means that now we can have additional genes that haven't been associated with arthritis as highly ranked. So here we just show that by combining features from the three data sets, we get improvement. So the transcriptomics in general works really well, but also those other sources of information give you additional added value to get the best results. And as I mentioned, you can easily test other types of classifiers and parameter settings to see which ones give you the best predictions. And here is just comparisons to some other machine learning algorithms that are available in those libraries. And also you can compare it to scrambled data. So at the end, you get ranked list of potential genes and targets that can be associated with each of those pain types. And you can see that those are different for different pain types. For example, a migraine have a lot of ion channels that are predicted to be associated with that pain type, while RA is mostly enriched with G proteins. So those are potential family of druggable genes that have a more representation in the top ranks of for each of those pain types. So now if I just focus on rheumatoid arthritis, if I just look at all of the genes across, regardless of any filter, this gene that encodes extracellular ligand, CCL4L2, is ranked #1, and it's a really understudied gene. There are only 7 publications, and none of them are mentioned to have anything to do with rheumatoid arthritis. So one of the things that we can do, we can now make predictions about what is this gene doing to cells, and what is the function of that gene based on the methods that I showed you before. And the other way to look at it is also to filter by druggable genes or druggable targets. And Debbie Shackman is also very interested in those type of molecules that you can target. For example, GPCRs and kinases and ion channels. So this is just looking at GPCRs, ion channels, and kinases, and you can see that GPCRs are overrepresented, like I showed you before. And if we look at some of those molecules, they're already known to have something to do with RA based on PubMed search. Some of them were already in the original, like when you see this known pain type, meaning that they were already in the list that Kathleen assembled for RA. But this third one on the list that had very high probability, GPR 132, has not been previously known to have anything to do with RA. It has only 30 publications. So it's orphan GPCR that is very interesting to see that, maybe it is an interesting target moving forward. So those methods give you hypotheses for testing. And this is the GPR 132 landing page of Arches 4, and it shows you that it has been knocked out in mouse already. And these are the predicted mouse phenotypes. And you can see that we predict based on gene co-expression relatively well the mouse phenotypes, and we can tell that it is an immune system related gene. And if you knock it down, you get some defects in the immune system, which matches like RA in general, that we know it's an inflammatory type of response that in the joints that cause pain. When we look at the CCL4L2 gene, we can see that it's also associated with inflammation and pain. For example, a CNS inflammation and abnormal immune system when you knock it out in a mouse. And this gene has not been knocked out in a mouse, so we don't have anything to compare to. So we do not have like this RC curve because it hasn't been knocked out in a mouse, and we don't have the recorded phenotypes based on the whoever provide that data, typically the Jackson lab or other places that knock out single genes and then report those phenotypes systematically. So this is a candidate, especially because it's extracellular ligand, to be a candidate to either have it increase or decrease with hope that it will affect arthritis positively. So we can potentially use those candidates as targets directly. But the other thing that we can try is to find the small molecules that can potentially increase or decrease the expression of those targets. And in order to do that, we have this new app that is an Aperture, where you can put in your gene, and then using the newest version of the Lynx L1000 data, transformed through another deep learning model that expanded to cover the entire genome, you can find small molecules that can potentially either increase or decrease the expression of the target gene. So if you go to this URL and you type in any human gene, what you get is those volcano plots. But those volcano plots, instead of visualizing like typical volcano plots, the changes in expression of a gene, it finds all of the signatures that maximally either increase or decrease the expression of the gene. So for example, for GPR132, which was one of our targets, we find this dinaciclib as a drug that potentially increases its expression. And then at the same time, we have tamoxifen as a drug that decreased the expression of CC4L2. And we already know that tamoxifen, one of the most common and severe side effects of this cancer drug, is inducing arthritis. While dinaciclib, although it's been prescribed, it's a CDK inhibitor, that although it was prescribing for different types of leukemias, it is been suggested to also have a positive effect on arthritis. So in both cases, it looks like if we increase the expression and activity of those two genes, we will have a favorable phenotype. And if we do decrease them, we potentially will cause arthritis. So this is an Aperture that was developed, and this is the results. The results also are available through tables that you can view and download. So a little bit about what is an Aperture. So one of the things, we have developed a lot of bioinformatics tools over the years, and those tools are relatively difficult to maintain and also develop. Those are full stack web-based applications that require some serious skills to bring up and maintain and also develop. So the Aperture is sort of like, it's a little bit of a shortcut or easy way to develop those tools. And basically what the way it works, you take a Jupyter notebook, which is, I think the audience here all know about Jupyter notebooks and how they're a way to bring in code and markup text and potentially interactive figures and data all into one place. And then you can execute those notebooks to as a part of your bioinformatics workflows. There are also ways to automatically execute those notebooks. And the Apertures, what Daniel Clark from our lab, the person that I mentioned, and has ties to Brazil, is a half Brazilian, and he came last year to Sao Paulo to present, developed this system where you insert into your notebook some code that converts the global variables in the notebook into a web-based form. And then once you compile the notebook with this software development kit, you get a fully executable web-based application that is Dockerized. And then you can deploy that application as a form like I showed you before. And then once the user fill in the form, for example, put in a Gname or upload their CSV file, the form is executed in the cloud, and then you get the executed notebook as a report. So a biologist doesn't need to have any, doesn't need to know how to run the code, but you can also download the notebook with all of the source code, and then you can execute it and modify it locally if you like. So far we've developed almost 100 Apertures, and they're all served on this Apertures catalog. And we published this paper earlier in the year in the journal Patterns. And it's been a pretty great success that now we can maintain and develop bioinformatics apps very fast for very specific tasks. And we can run those workflows in a way that does not require any coding. And it's very modular. And then that's something that I want to highlight here. So I'm just going to summarize all of the resources that I went through. I went through pretty fast. So hopefully we'll have some discussion questions to clarify and potentially get into more details. But first I showed you this gene and drug aggregator. So this idea that there is all of those landing pages, and then biologists, they really want to find information about their gene of interest. And all of that information is now spread out across many websites. And some of those websites, they are also aggregators, but this website simply aggregates the websites. So we've created that and we haven't published it yet. And then I'm moved on to talk about Harmonizome, which is an aggregator, but it's also doing some processing to the data to make it harmonized and easy to integrate. And the most exciting part is that it makes it ready for machine learning. And Enricher is sort of like a specialized version of Harmonizome, where everything is organized into Genesets, and it's a Geneset search engine. Both Enricher and Harmonizome have been a huge success for user perspective. We have, I think, over 2,000 users each day that accessing those resources. And then I moved on to talk about Arches 4. And Arches 4 is also a great resource on its own, just providing all of the data in geo as in a uniformly processed format. And also with Arches 4, you can build those gene-gene correlation networks and make predictions about gene function. And it works really well to predict gene function in a very simplified way and illuminate knowledge about understudied genes. And then I showed how we can do this same type of thing for drugs. So I mentioned this way of predicting side effects with this SCP-L1000. And then I also showed drug Monizome as a version of Harmonizome that is for drugs. And then I showed you some of those Apertures, and then how we are moving the development of bioinformatics tools into Apertures and the Aperture catalog. And specifically, once you have identified some targets, you can use this particular Aperture to find drugs that would increase or decrease the expression of your target. So just the final slide is an acknowledgement for all of the lab members, and all of them contributed to all of those resources that you've seen. So I'm very grateful for all of their work, as well as the funders. We are funded by 3 data coordination center efforts, the IDG, the LINKS DCIC, and the CFDE. And also we are in the data coordination center for the Lyme consortium that is privately funded by the Cohen Foundation. So that's it. I will now.
Thank you so much, Dr. Avi. So it's really impressed, all these tools. I thought not less that you're gonna show some really cool stuff, but I confess that I'm kind of disappointed, because I thought you're gonna mention some tools related to virtual reality, meta, because that's the. So are you planning something like this, like completely, like that use Oculus Rift, this kind of stuff? That's just a curiosity. I thought you would be already in this level. I'm just kidding.
No, I think it's a good idea, and we have tried to consider, 3D network visualizations and VR. We are looking at like hand movement, control of the mouse or whatever, or the interacting with the data. But at the end of the day, those things, they are still, I don't think we are ready for them yet.
That's true.
So I feel like the technology is not there.
It's just entertaining, right? For now.
For now, it's more. It's not practical, and it doesn't. It's not gonna really make you increase your ability to understand the data.
That's true.
So yeah, but we are open to it. If there is a good ideas and there is a way to do it, and it's one of the challenges is also the coding of that is still not there. It's not that easy to use the libraries, and it can become cumbersome. The browser may be too slow. So it's gonna happen, but I apologize for the.
No, I was just kidding. Of course, it was really impressed. So I'm wondering, like, instead of the aggregators that you showed, the kind of tools that you showed, that where you aggregate a lot of database and tools into one, are you guys also making an effort to make a consolidator, to try to find consistent? So for example, in Enricher, we use a lot in Enricher, and then we find the same pathway in different database, but it would be nice to have like a tool that tell us that, look, when we look at all these database, this is the pathway that appears with different names, but they are actually the same in several of them, so instead of looking on each one of the database, that's the kind of stuff I think could be even faster than that.
Definitely, that's something that's an issue. One of the things that we are trying, so one of the challenges with this is that a lot of those gene sets, they're degenerate. So it's hard to decide where is the cutoff of saying, okay, this is this pathway. Yeah. So there is a little bit of overlap with all of those pathways. And we had several projects that are still like, haven't been very successful because it's such a challenge to figure out, what is the relationship between those gene sets. And one idea is that you have that small set of modules, or at least what I was thinking is that there are some genes that go together all the time. So maybe those are like words that in a gene set, they are all coming together in one way. And then maybe in another gene set, they have some different words. And we tried to look into it, and it's just messy. It's complicated. It's not as clean a puzzle. But I think what you're talking about is a really great puzzle to solve, and the information is there. It's just a matter of like putting it together. I know that MSigDB has done some work to do this, where they created those canonical gene sets that I don't remember the name of it, but they have 50 gene sets that's supposed to cover everything. But it's challenging, and it's not as trivial as it seems, but it is definitely something on our mind that we keep trying to do.
That's awesome. So there are two technical questions in the Zoom. People from YouTube, you can also ask questions. So Tomás and Mauro ask if the RNA-seq data, gene counts, and also the transcript level counts tables are available for downloading the Archie S4 website, or if it's in your server without access.
No, it's all available on the download page on the Archie S4 website. So if you Google it and then go to download, you can get those tables.
But can you get all the 900,000 tables, or it's more like a one by one? Really? Okay, cool. So Ellison from YouTube is asking, I'm going to translate that he would like to know, how did you handle the number of features when you use the machine learning to deal with complex data? I'm going to complement his question that did you use also feature selection methods, or was purely machine learning?
Yes, so we do different types of feature selection in that example with biochemistry, the biochemistry paper about the pain targets. We use PCA to reduce the similarity matrices. So we created similarity matrices first from the gene and protein and Go terms, and then we reduced that to 200 features based on like a PCA applied to that matrix. But there's many other ways to do it. And it is important because once you have so many features, you have to do it.
Cool. So there is another question about for example, the KEGG pathway for rheumatoid arthritis. How this pathway is related to the top 20 genes that you found associated with arthritis.
Okay.
I was asking that because I like making pathways also when they are not in KEGG. In our group, we try to build them, and I saw that there is one there related to dendritic cells and etc., more related to immunology. Do you think that it might connect to the genes that you labeled there?
So the way we do it is we just compute the average correlation based on gene expression.
I know.
And if there is, and then we rank the terms. So the correlations, the average correlation will not necessarily be big, but if you just rank the terms, you'll be able to see which terms are most relevant for that gene. And then, so in principle, it's only for one gene right now. So we just look at each gene and then how much it's correlated with the KEGG pathway. And so the top 21 that we said that are rheumatoid arthritis, we can tell you the KEGG pathways that they may be related to.
But what I am wondering is if those 20 genes are either connected to that pathway or maybe they have a new scenario. I mean, a pathway that is not there, that one has to build, you know?
Yes, I think it's a great suggestion, and I think that it's also related to the comment that Elder made about the bringing all these data sets together. You can discover novel pathways.
Yeah.
Yeah, but we have not done that. But it's the information is there because it's all omics covers the entire genome. There must be some novel pathways that are just waiting for discovery.
So now it's completely my curiosity, Avi. Where did you get the ideas for the tools, like the inspiration? Are you watching a seminar and then say like, look, I'm going to solve this problem? Or is a postdoc that brings this to you and then you decide, let's develop a tool for this, or it's you doing the analysis and then say like, oh, this is bad. Usually how it is the process.
We do have every 3 weeks, we have a brainstorm meeting where every person has to come up with an idea for a project. And then we have a prize for the winner. So people are really trying hard to come up with some cool ideas. But at the end of the day, I'm making the decision of like what we will push forward. And then, but it is, sometimes, people come up with good ideas. Sometimes it comes from me. I can't really trace it back. I don't want to take credit for anything that I shouldn't be taking credit of. But it's just those ideas come from multiple sources, I guess.
Wow, that you've just got me an idea for running this prize context for ideas, and I hope this list is a very long list that you're still gonna implement. So Avi, I thank you so much for showing this nice
The question.
Oh, you have a question?
Question.
Sorry,
no problem. Thank you very much. Thank you very much, Avi. It was a really cool presentation. We are familiar with some of your tools, so we've used in the past Enricher, and it's just amazing. I really like the idea of the aggregator, actually. I was just wondering, what do the owners of the platforms that you extract data from, they think about the licensing? So how do you deal with the licensing to use their resources within your infrastructure? What's your strategy?
So most of them, we never really receive a complaint of somebody telling us to take something down. Many of them have restrictive licenses where we cannot resell the data. So there is also a lot of commercial interest with companies that want to use those tools, but we can't really sell those tools or license those tools because a lot of those tools rely on databases that require a license to make profit. So everything is open and free.
Yeah.
And then
thank you.
and then in some cases, especially things that we are developed from scratch and things that are completely open, then we can license those parts. But for the issue is for most of those resources, they make it free for academic, and if you are a company, you have to pay.
Yeah, great. Yeah, and it actually goes to my second question. So in your team and the people that passed by your laboratory in the last years, what is the career pathway? Are they becoming like academic, most of them, or they're going to the industry? So what is the tendency on your group?
So it depends. I guess the majority actually went to industry, and they are having like high positions in companies, but there are several people that they continue in academia and they have their own labs. Yeah. So it's a little bit of a diverse, but it's not a typical path. So there are different paths. Yeah.
And how is I'm sure that it's New York City, so it should be much more exciting than other places in terms of selling your idea and getting to a pitch to establish a startup. So can you like explain for the audience a little bit of this aspect of leaving your laboratory with all the skills you learn, and then maybe try to pitch some venture capitalists who found the company. So what is the experience of your colleagues leaving your laboratory?
I think I did not have anyone that actually tried to do that yet, but there is Mount Sinai. I have this, the lawyers and the Office of Business Development, that they're pretty strict on how much you can take with you. So I think that's maybe a challenge for some of those people that are graduating from the lab. And in general, I'm interested in getting into it, but we never really got to do it. So there is a lot of potential there, because you can see how we can very quickly discover targets and drugs for any disease. But we have not really capitalized on pursuing that as a startup yet.
Okay, thank you. Do we still have time?
Well,
Yes.
is there anyone?
Go for it if you.
Yeah, I have
wanna
another
open your
question. It's
camera.
more, it's a question more regarding really the resources that you presented. So you said that you try to pick up new genes that are not associated with diseases. So and then you explained as pain as a use case. So are you extrapolating the information of the new genes that you are picking up to the metabolic context? I mean, not metabolic, but to the protein-protein interaction world, to see whether those genes are part of the same complexes, or if they interact like binary informations, like coming from the PPI world.
Yes. So in the Arches 4 paper, we showed that we can also predict PPIs based on co-expression. And this is also relates to Miguel's question, where you can potentially start identifying interactions and pathways based on those predictions. So that's definitely a way to also go about it. And it's not a major way. So there is still potential for using all of the combined data sets for PPI predictions.
You know that Adriano has a tool that does the connections by text mining. So we are used to take the tool to create original pathways. That was my idea of trying to connect your 20 genes into pathways using text mining.
Because you demanded also literature information when you showed your table, saying that some of the genes are not really mentioned in the publications in terms of having papers associated with. So the tool could somehow find this kind of text mining associations and then try to put it into context in a metabolic perspective, what your genes are doing, the new genes are doing.
Yes, sometimes you can figure out genes that are regulating genes that are regulating genes, and then it connects to the other one in your list. Somehow.
Nice. And the final one from my side, at least, is regarding the knowledge graphs that you showed. So you said that at the beginning, you capture information from different sources in order to represent the knowledge as a major knowledge graph. And what do you think about overlaying different types of knowledge graphs that exist out there, such as PharmKB or OpenBioLink, for example, and putting them as a major resource in the same way that you do with the gene expression information, for example? So in this way, you could somehow aggregate different knowledge graphs in order to answer your questions.
Yeah, I think it's a good idea, but I'm a little worried about the size of knowledge graphs in general. Like they can become really, really, really big. And then it's very hard to find stuff once they get to like billions of nodes. So there must be some other way to reduce their size or find a way to filter things so they don't become unwieldy.
Okay. Thank you. Thank you very much. It was a very nice talk.
Thank you.
So now that you open again for questions, you mentioned about the gene pain gene pain paper, right? That is pain is a generic symptom. Are you thinking in also working for some gene fever paper? Because we also noticed that several things can cause fever, and that is interesting for us that work with infectious disease. So I don't know if someone in your group is trying to do the same framework for fever.
No, but it sounds like a really good one. Right now, I know that fever, like people don't really understand how it happens. So, and it does pop up. So I think it's a primed question, but we are not pursuing that particular one. So you should go for it.
Yeah, I won my prize. Cool. Okay, so I guess there's no more questions, and I would like to thank you so much for your time and everyone for watching. Thank you, and I hope one day you're gonna come to Brazil.
Yes, I'm looking forward
Yes.
to that. Okay.
Thanks a lot.
Thank you so much.
Thank you very much.
Thank you. It was a beautiful class.